Hi. Hello, everyone.
Hey, Simon. Thanks for
Yeah,
joining us.
thank you for having me.
Yeah. So we'll go ahead and get started. Um, again, Simon, thanks for carving time out of your day. All the participants, again, thank you as well for joining us this morning. Um, just wanted to start out by introducing myself to and who's going to be on here today. My name is David Fleck, and I'm the founder and managing partner of FreeFlow. And I'm joined today by my partner Kevin Barrett, who is a managing partner as well at the firm, and also Simon, who is the CEO of Mango. At FreeFlow, just we invest in things that are basically at the intersection of what I like to call atoms and bits. So that's the intersection, basically what we describe as AI plus X. So it's compute plus other scientific disciplines, essentially deep tech enabled by AI. And I think Mango is just a wonderful example of that. We focus on specific ecosystems at the firm. So we find that there's interesting network effects by focusing on Caltech and then now Berkeley in terms of just where we deploy. And we'll mindfully and thoughtfully expand our own model. But Mango came out of the lab of Dr. Yang at Caltech, just a wonderful kind of polymath at Caltech, and he's a specialist in AI, but specifically in computer vision. And so, computer vision is an interesting space where you're basically using AI, but in the visual sense. And so the reason we invested in Mango this morning, I was just rereading our initial deal memo into the company. So I always like to kind of revisit why we invested, and actually you're executing on exactly. I mean, I don't think I've shown that to you, but I'm happy to. But you're executing on exactly what we thought would be the opportunity, which is the fact that the in biomanufacturing today, the Petri dish, which is a 19th century technology, is still the gold standard, because you have to check for biosterility and bioburden and those types of things. And the Petri dish just takes a lot of patience. It takes a lot of time, hours or even days, or sorry, days or even weeks sometimes, depending on the application. And then Mango, by using computer vision, can actually, and Simon will walk through this, like get much more timely responsiveness in terms of how the batches of biomanufacturing are progressing in terms of contaminants and things like that, which can lead to just a much more efficient process, number one. And then even perhaps more importantly, at least to me, is the fact that it even opens up novel therapeutics, because the testing lag time actually makes it impossible to have certain types of therapeutics today. But by increasing the or decreasing the time, increasing the efficiency, you can actually attack some of those problems. So Simon's going to talk about their initial market and just the company himself, the big vision. So just wanted to say thank you again. Kevin and I will probably disappear here and let Simon kind of do some, introduce himself in the company. And just a reminder to everybody that you can go ahead and put questions into the chat feature here, and then we'll get to those along the way or at the end as well. So this is meant to be a conversation. So with that, I'll turn it over to Simon.
Thank you. Appreciate it, David. And thank you, FreeFlow, for having me today. And super excited to talk about Mango and kind of what we see as the future. So I'll go ahead and share a little presentation. Okay, perfect. What we want to talk about today is really like Mango as a company, but also what the vision for Mango is and what we see as the future of micro QC. And micro QC stands for microbial quality control. And it's, as I also go into a little bit deeper, it's a big part of a lot of different industries, but especially in the pharmaceutical industry, it is a very crucial area that ensures that drug products are safe for consumption. So this is a crucial point. So what are we trying to do at Mango? So Mango, the big vision is to become the definitive micro QC platform. And micro QC consists of a couple of different things, but the two main tests are what it's called bioburden and sterility. And for bioburden, you count the number of either fungus or bacteria in a sample, and you enumerate the number, and so you spit out a number. That's the number you want to hit. You want to be below a certain threshold. And for sterility, you want to make sure there's no presence of foreign microorganisms. So those are the two main tests, and I'll kind of delve into how we go from those tests to become the micro QC platform. And as everyone here is probably aware, it really starts with having a great team. And this is not my first. It's not my first startup. I'm, as the CEO of Mango, fourth time founder or CEO of companies, and I'm happy to say that this has been the smoothest ride to date, and I think a lot of it that I attribute to the team and having had like really incredible colleagues in the past that I was able to recruit from this team. So my previous startup was acquired by Snap, so I have a lot of colleagues from Snap. Snap is the parent company of Snapchat, and if people might not be aware that Snap actually has a whole Snap lab, which employs hundreds of engineers working on hardware and really hard problems around that. And on our team currently, we have 7 people who are from Snap. So it's a very different discipline that they come from, but it's really they recruited because they were the best of the best. They're the best people I knew. And when you put really smart people, really capable people on hard problems, good things tend to happen. And that was the philosophy we took building up Mango, more so than getting people who were traditional from the industry. We really sought to find people who could solve these problems with novel ideas and use novel ways. And a lot of this also we'll talk about ties into using the latest computer vision and AI and machine learning to really unlock a lot of value. And to talk a little bit about what the problem is that we're solving. So right now we have a lot of QC tests. There's like tens of thousands of tests done at each lab every year. And so it's a lot of high volume tests, but this process is very manual. And as David also said, this is really from like, the 1800s technology. That's when the Petri dish was first used, and 99% of tests today are still done with a Petri dish. So not much has changed like in the last 175 years, which is kind of crazy to think of. This means that every day, someone has to go in and check incubators, manually check a Petri dish and look at it and see if there's growth on a Petri dish, noted in a notebook. For most of the labs that we deal with, it's actually still on pen and paper. This is how it's inputted. And this is part of like the FDA mandated test that has to be done to make sure that the products that are being made are safe. And this is everything from vaccines to pills to everything else in the pharmaceutical industry that is consumed is pretty much undergo these kind of tests. Even ointments and those kind of things also undergo tests regularly. And in terms of the process, it's not like a single test. Bioburden and sterility is done throughout the manufacturing process because you really want to make sure that all the ingredients going into these tests are not contaminated, because once you have a contamination event, it is very expensive. And yeah, and now I'm going to like how expensive that really is. And it's not only is there a hold time for these tests to clear before they can be released to the public, but a big part of this is also that each event, when you have a contamination event, is extremely expensive because you have to shut down the line, you have to clean all the parts. So we have learned that it can cost over $10 million to shut down a line in terms of lost revenue and cleaning the whole system, depending on how bad the contamination is. So how is Mango actually solving this problem? So this problem, we are attacking it by providing an instrument. And this instrument is here where we see the M6. It has 6 individual bays. So it can run 6 tests simultaneously. And each test includes an incubator and an analysis sensor that will actually give a result of how many CFUs, that's colony forming units, are in a sample. So it will spit back a number and let you and also determine whether that sample is contaminated or not. And in terms of the cartridge itself that goes in, is really our version of the Petri dish. So the little square puck that you can see is our cartridge that it holds the same agar you would find in a normal Petri dish. So in terms of the materials used is identical pretty much to what you have in a normal Petri dish, but it's a small version that we have enhanced in order to give us faster results. And I'll dive into that in just a second. So here on the right side, you will see this is the traditional Petri dish. And what you actually see here is millions of cells. So you are not seeing a single cell. It might look like a tiny dot, but even these tiny dots are millions of cells. So this is why it takes a long time for you manually count these. And not only do you have to count it with one person, you actually need a second analyst to go in and verify the results as well, which is part of the slow and labor-intensive process. What we have in Mango is, like I said, our own cartridge, and this is a one-time use cartridge. This cartridge we're able to detect down to a single cell resolution. So the technology is able to go down into like almost a half a micron, 500 nanometer scale resolution, but typically we have it about like 1 micron resolution. So we can see single cells actually forming. And the way I'll talk a little bit about the technology because it came from Caltech originally out of Dr. Yang's lab, as David mentioned, and it was really, really an ingenious solution to a problem where you have a limited amount of resolution, but by shining different LEDs from above at different angles and having all that known, you can actually you can think of it almost as you break down individual pixels into smaller pixels and get higher resolution. And you do this by algorithm and computation. You don't do it by physically breaking down the pixels. So you get a lot of resolution even with a high resolution sensor. So you have each pixel on our image sensor is roughly 5 micron, but because of the algorithm out of Caltech that's called ePetri, we are able to see down to a single micron resolution. So these are some of the kind of things that got me really excited when we first started looking at the technology and talking about with the different pharmaceutical companies. What is kind of like the trends that makes this a good timing? So we have one thing is this is a big bottleneck in QC. They're actively looking for solutions, and this is pretty much across the board for pharmaceutical companies. They're looking for what we call rapid bioburden and sterility. So they're looking for faster methods since it is a bottleneck that's costing them money. And the second part is they have a goal of lights out manufacturing. And this is, whether the goal is 2030 or 2035, it sometimes shifts from manufacturer to manufacturer, but we have consistently heard that they want this to be lights out manufacturing. And what does that mean? It means that on a production floor, there's no human interaction. There's no humans that really can mess up test and cause contamination. And this is where the biggest source of contamination right now is really from humans making mistakes. So they want to take as many people out of the loop as possible and have this be able to run the whole process without any human intervention. And the third thing that is very interesting is the gene and cell therapy space. This is a fast-growing space. It's a very interesting space because it's really taking the human cells, modifying them to attack. In many cases, like we use it for blood cancer, so you attack the cancer, and it's really, really effective, but it's also very expensive. And one of the biggest problems is that there currently isn't a good sterility method that is faster than the Petri dish. So you currently have to inject patients at risk, and if you're at risk, that means you have to give them additional drugs to fight things that might not be there, which is of course not great for patients. And with this technology, we can really avoid that. And so I'm super excited about how Mango can help in that sense as well. Talk a little bit about like the technology itself and why I think this is super interesting. This especially ties into, AI is being talked about a lot, and for a lot of things you talk about like big language models, but this is really a different type of AI where we use the visual images to run our machine learning algorithm across it, and we have trained this on thousands of data points that we already collected, and humans for the first data set actually sit and manually tell the algorithm what a CFU is, what is a bacteria, what is a fungus growth on each of these images. So we've done this with our scientists for the last 2.5 years now. We've been training these algorithms, and then we can use that to give us very, very good data, very accurate data. And I'll show you some of the machine learning actually working, but where we are now, it's pretty much on par with a human scientist that has been trained. But as for certain things, and in the future, we expect it to be significantly better than a human to be able to pick up any growth or any CFUs. And one of the big key things here that I want to emphasize is that it's not enough that you have the AI and machine learning models to be able to detect this. You really need the data to train these models. You need the data to also have something to analyze. And data is, when it comes to machine learning, it's very fickle in the sense that the quality of the data is extremely important. So this is why a big part of why we have such a moat in this space is that we're building the hardware that captures the data. We had other companies approach us several times to ask if they could have access to our data, if they could use our data to run their detection algorithm. But this is really a big part of what we have that is special. So we have the hardware that's very consistent and makes it ideal for machine learning and AI to not only do the learning, but also the subsequent testing. So we're starting here with bioburden. This is the product that is coming out here. Actually, we shipping our first products to. We have 3 units shipping here in April, and this is in the bioburden space. So they will be used for bioburden testing, and then we are going into sterility, which sterility is very much related to bioburden in many cases. Sterility, we're using the exact same instrument, we're just changing the cartridges slightly. So instead of having 1 test for bioburden, you have 3 tests for sterility, 2 at different aerobic that are different temperatures, and 1 anaerobic. So currently we're in the R&D phase on the anaerobic cartridge that goes into sterility. And from there, we really have different what I call verticals that we will attack internally in terms of R&D. We have automation, and it really starts with the partial automation that we already implemented. So currently, all you do is you insert the cartridge, it runs the test, it spits out the numbers directly to the LIMS system, and then you get it back, the cartridge, you discard it. So that portion is already automated, but we want to enhance the automation. So we're doing filtration, we're automating that portion, and we are automating different levels. So we have a roadmap for how we're going to achieve this full automation that eventually will lead us not to only add line testing. So this means that you can pull a sample right at the line of where you're manufacturing the drug product and run the sample right away. We also have patented technology that involves actually inline filtration. So that is something that we're super excited about as well. The other portions that we will optimize is really like for the cost of each cartridge, and we already have line of sight to get this cost much lower and actually comparable or even lower than running a traditional Petri dish. Currently, there's a premium to our cartridges, but you get the solution in a matter of hours rather than days. I'll show you the data in a second. And then we have the third portion, which is really building out the software. And what we do with the bioburden and sterility, those two tests are really the cornerstone or like the foundation of any QC lab. So having those pieces, we tie into their LIMS system, the manufacturer's LIMS system, and this is a system that holds all the data throughout the manufacturing process. Setting up those ties is burdensome, as you can imagine, working with IT and getting all the information flowing correctly. From a technology standpoint, it's not that necessarily that hard, but from an implementation standpoint, it is quite a pull. So having those highways already with all the information flowing really enables us to offer other software solutions that normally would not necessitate having this LIMS integration. But because we already have the integration, it's very easy for us to add on services. And then finally, we talk about market-specific hardware, and this is where we have a lot of opportunity too. We have in the cartridges right now have TSA, which is a very common, the most common type of nutrient growth media. But in the future, we also looking to develop things that are more specific for fungus, and all. We have other types of cartridges that we're currently developing that will open up the market even further. And that really, those categories really bring us into having a full micro QC platform. So we want to be the definitive platform that is used doing testing in the pharmaceutical industry. And then as you go below the micro QC platform, what is really interesting here too is this is for pharma. We talk only about pharma so far, and pharma is definitely our first very targeted group that we're going after, but there's so many other industries that actually also require this. So we have food and beverage, we're talking about cosmetics, we're talking about even things like baby wipes need testing. There's a lot of different industries that need this testing that we will also have the opportunity to go after in the future once we kind of established our micro QC platform. And this is some of the tests that came out of one of the big pharmaceutical partners. It's a top 5 pharmaceutical company that tested the Mango instruments in their lab. And as you see on the left side, this data is not captured by us. It was in the Brazilian Journal of Microbiology, and it shows the conventional method, which is the Petri dish. And the Petri dish, the time to detection was anywhere between 48 to 96 hours for these 6 microorganisms. And these 6 microorganisms are very representative of kind of like the swath of typical organisms that you will see. The bacterial alert is currently the gold standard, or like the fastest alternative method. It was significantly better than the conventional, but it's nowhere near what Mango was able to deliver with under the same conditions. So the Mango results were within hours, not days. And here I actually want to show you guys some video of how it works. So this is the surface of our Petri dish that is imaged. And here, this is done at a normal resolution. You can't see any microorganisms, but if you turn on the overlay with the AI, it actually identifies where these organisms are. And if you manually then zoom in to the area where it is, you can see it is impossible for the naked eye to know there's anything here. And this is what we call like the raw images. So this is before the whole caltech processing has occurred. And if you turn on the super resolution, all of a sudden you can actually see there is a colony forming unit. So you can see there's growth. And because of our resolution, we can actually keep zooming in, and we take these time steps every 20 minutes so we can see the growth. So here you actually see how the colonies are growing from is almost a single cell here into like bigger colonies. And the machine learning is then able to count these. And on the next one, what I want to show is this is the machine learning overlay. So this is the machine learning actually identifying not only with the color of the purple, it identifies what the CFU is. It also identifies with the blue where it originates from. And on the left side of the screen, you can see there's actually 4 different colonies that originated. Under normal circumstances, these might count as one, and because they have grown into each other, and before you can actually detect them. But on our system, we can see that these are actually 4 distinct colonies that are growing, and the machine learning then captures this data. And on the bottom right side, you can see this is how the machine learning is operating and counting these as time goes on. And this is 12 hours into the sample. Each of these blue squares are the dots, or like the time steps at which we capture this. And here on the left side, you can see this is a small area of the larger surface that we are actually looking at right now. And because of the resolution, this is hard to see with the naked eye. This is something humans have a hard time because the contrast is not very high. And finally, what I want to mention on this that is super impressive is that this is actually through a layer of T cells. So before we talked about gene and cell therapy and how that is a very hot thing at the moment and fast growing and very effective. Normally, you would do this on a petri dish, and we didn't actually know before we tested this at one of our pharmaceutical partners' labs whether we could do this, but we're able to image the growth of bacteria and fungus through a layer of T cells. So this has, I believe this one has 3 million cells. So it has filtered 3 million cells, and that's spiked with E. coli. So this E. coli is then growing among the T cells, and we can then image this. So currently, we're very excited to come up with this here at the end of the year. And here's some of the other tests that we've done in T cells background. And this is very much what we're doing right now in terms of R&D. This is our focus is to get the sterility solution out here by the end of the year. And that kind of ties into where we are right now. So we've done a lot of proof of concepts. We had the pre-production units that we shipped here at the end of the year last year. We shipped these to 4 of our pharmaceutical partners. They have them currently in their labs, or they're finished testing in the lab with the M6 pre-production units. And then here in April, we'll be shipping our first full GMP units. We're very excited about shipping the first 3 units here in April. That would then undergo a full testing to become part of the production environment. As we head to the end of the year, what we're currently focused on in terms of R&D is having a sterility solution specifically targeted at the gene and cell therapy industry, because they're very much need of this and are willing to adopt it very quickly. So that is our first target, and we have already had commitment from one of our partners to start testing before the end of the year. So that will lead us into Q1 of 27, rolling it out to more partners and roll it out into the market. And that is all for me for right now, but I'm happy to discuss and take questions.
Thanks. Excuse me. Thank you, Simon. That's
it's just really cool to see it come together the way that it is. Excuse me. One of the things I wanted to, or that I always like to start off is just asking people or asking, CEOs like why did you want to get into doing this? You could do so many different things, and there's usually an interesting journey or story to get at that question. So,
Yeah. I wasn't actually looking to get into something else. I'd already, in principle, agreed to take on a CEO position at a startup I pitched to Porsche. The car manufacturer is a very different area. But when I heard about Mango, it was one of those opportunities. It was one of those technologies where it made so much sense, and I really couldn't turn it down. I felt you once in a. I feel like you get these opportunities very few times in your life,
mm,
and this is an opportunity to really change an industry, to really not only change the industry and make it better, but also make it better for humanity. That was a big
mm,
part of my drive, that you can actually help people. You can get tests much faster. You can help these advanced like therapies. People with late stage cancer can get treatment faster. It felt like the right thing to do, and this is no knock at my previous companies and the great partners I've had there, but it was just not. It didn't feel like as consequential in the past. It didn't feel like um, as important as this is. So this is really what drove me to do this, and yeah, I'm just as excited as when I started, if not more. I really see just how much the team has done to get this up and running. And yeah, maybe we can talk about that as well, like how the team is from a different industry.
yeah, yeah, yeah. That might be. And if you want, you can probably take off the
Yeah.
chair unless you want to
Uh,
bring it up for specific questions. But yeah, the team, that's an interesting. I love it because our or even my own background. I was a product manager at Google, and then I'm now doing deep tech and AI essentially, which is like a step in a different or it's a continuation maybe of the journey. But either way, back to you and the team. I think it's interesting to see how you obviously have to have subject matter experts that are guiding strategy, and
yeah.
because this is a very technical space, but you're also able to bring in people that are just super smart at specific like computer vision or whatever the example may be. Maybe if you would just talk more about that, I think that's
Yeah, yeah.
interesting.
So the way the company is kind of divided, and it's not, we really have like different groups of expertise. So we have, of course, the microbiology side, and the people on that team, they do not come from Snap. They are very much like,
Mhm.
hardcore scientists, and this is what they studied, and this is what they love. And that's how it should be. And the same with sales. It's very much people coming from the industry. We wanted that veteran experience in those areas. But when it comes to both the hardware and the software and the machine learning and AI, this is really where we look to recruit the best of the best, and not necessarily people who came from the biotech industry specifically. So I went after the best engineers that I knew, and very happy that, yeah, seven of us from Snap are a part of this company. And one of the things I wanted to mention is that what we saw, I've seen the previous attempts, is that you can have very smart people. You can have people with, you know, going from the best schools, the best, have done the most interesting research in the past, but not being able to then transform that into actual products that are working reliably. And I think when it comes to commercializing a product, what you really need is great fundamental engineering skills. So this is something that's very much more industry agnostic than you would think it is, even for something like biotech.
Yeah. One thing that, and Kevin, please chime in too with any questions you have as well. One question that I think might be helpful, and I see some of the other questions popping in too, which we'll get to. So thanks for those. Please continue putting those in there, audience members. But is the business model actually is where I was going with my question is, my as we see it develop, it seems like it, and this happens in deep tech. It takes, you have to de-risk it. You have to do the pilots.
Yeah,
But then once you get in there, it seems like it's pretty, I'm leading the witness here a little bit, but pretty sticky, right? And then you
for
have
sure.
you have a few different ways you can potentially, or that you're planning to monetize those.
Yeah,
Razor blade analogy and stuff. So maybe just unpack that a little bit, just so everyone is tracking with the model and and I know you touched on this, but just like I think it's worth digging in on that, just so people understand how this might evolve over time.
yeah. So it is very much like a sticky model. Once you have to go through, different phases. So you have the feasibility phase, then you go into validation, and then you go into having into production. So there's, quite a bit of a effort that has to go to get through those phases with the different pharmaceutical companies. As the big ones, and I'm talking like the top 5 pharmaceutical companies that we're currently working with, once they've gone through validation, that will make it much easier for others to not have to go through that rigorous validation because others have already done the legwork. But this first part has taken some time, and we have only been working on it for 3 years, I should say. But it's once you're through that, you really have a long-term partner because they're buying these for like 10 years. They're not looking to switch these technologies every year. So for us, we sell them the instrument that is, $100,000, but then we have each cartridge, each sample is then, as you said, like a razor blade model where that's where we make our margin, and this is where we can keep selling them these cartridges on a monthly basis, but also add additional services on top of that. So software analysis services that we're planning on rolling out as well.
Yeah. So, David, I had a question for Simon, just relating to cell and gene therapies. Simon in particular, and you sort of made the point as part of your presentation that that's a particularly challenging area. And while cell and gene therapies represent a relatively small part of overall drug discovery and development today, it's growing in a very meaningful manner, estimated at 20 to 30% compound annual growth. So it's clearly moving in the direction of becoming a much more significant part of overall drug discovery and development. So could you sort of just walk us through what happens when you have a biomanufacturing line that's manufacturing a gene or cell therapy, and take the old technology in a petri dish where it could take 96 hours or longer to identify a contaminant versus minutes to hours, let's say hours, with Mango. Is the line still running during those 96 hours? And then what happens, how do you get to like not at the line item level, but that $10 million cost of shutting down a line, right? Of cost and lost revenue, just to sort of help us understand how that sort of comes into play with cell and gene therapies.
Yes. So cell and gene therapy I'm super bullish on this therapy. It makes so much sense to me that instead of giving like a generic treatment, it's a very like targeted specific treatment. And this is why I think the industry as a whole is so bullish on it, is that it's so effective, right? So even for late stage blood cancer patients, you see the look at the data, and the efficacy is just super high. And it's one of the best types of treatment that we can offer. But of course, as you mentioned, it is very expensive. What the cell and gene therapy process is a little bit different than the normal, like large scale pharmaceutical process. For the large scale pharmaceutical process, you have big batches that are like, could cost like $10 million each. For gene and cell therapy, it is much smaller. It's smaller batches. It's a smaller production facility, but we still have a process where you take the cells out of the patients. You then modify these cells to then go back and attack the cancer or the what you want to treat in this case. And you have to inject these same cells back into the patient. So there's a whole manufacturing process that goes through that. And in that process, you do take samples along the way. It takes several days. You take samples along the way to ensure that there's no contamination. And currently, as you mentioned, this is very slow. And it's actually so slow that by the time you have the drug or the cells ready to be injected back into the patients, you usually or you don't have an answer. And today, unfortunately, you cannot wait for that answer because not only is the patient sick and they need treatment as fast as possible, also the cells actually die off. So the sooner you can have the cells back into the body, the more effective it will be. So what they do today is they often inject the cells back into the patient with a cocktail mix of other drugs that is, of course, is not great for a patient. And you inject these drugs not knowing whether they're needed or not. And this is really where Mango comes in, and we can give this results much faster, so you avoid having to give a patient drugs that they don't need, that can have side effects that are not good for the patient.
Well, that's really helpful, and that's why managing around and decreasing adverse events, which these drug cocktails can help contribute to, is so important. So thanks for walking us through that.
Yeah, you have to admit. And it's very interesting. We even had like emergency room doctors reach out and ask about like how can this be used in emergency rooms? Because right now they get people with different infections and they have no idea what it is, right? Like, can they use this to know whether antibiotics is even effective? Because currently, I was talking to one of the head doctors at a big Norwegian hospital in Oslo, and he was saying that they are mandated to reduce the amount of antibiotics. So can they? So they have to wait several days to see if something is effective before they switch over to antibiotics. Can Mango's technology be used there? And I said, absolutely, it can. But this is kind of one of those verticals that are very interesting in the future, but we have to be laser focused on serving like the pharma production first.
Can you talk a bit more about maybe, because I know you're working with, and you can't talk about the names, but just it seems like, maybe to summarize it, large pharma companies are very excited about this technology.
Yes,
Um,
yeah.
and so it's like of the top five, there's just or of the top 20 or whatever, you're in talks with quite a few of them, which is great to see them actually excited about this technology and starting to adopt it. Can you talk to why, maybe like the driving force? Is it efficiency, or is it because they think they're actually, is it like expense and time to market, which I guess is revenue as well, but then, or is it more on we're going to be able to get new types of drugs through the process? Or is it both? And I'm just curious, like how are they looking at what you do and how it fits into strategy for
Yeah.
them?
So I think there's two main driving factors. I think one is, of course, cost. Right now, these contamination events do occur. It is expensive. They also spend a lot of money running these tests, sending them out, and it can cause delays in the production. So that is a cost aspect of it. I think also another big driver is they have a roadmap for how they want lights out manufacturing to be. And largely all these manufacturing processes, we talked to even we've heard this from several big companies, so I feel it's very credible that they have solutions to most of these pieces, but they don't have fast automated solutions when it comes to bioburden and sterility. So then that's kind of like a missing piece that where we will slot in very nicely. And I think just coming from a different industry in the robotics where the sales process is very much like you have to really convince them that they have a problem and that you have hit the right solution. This is very much a sales process where they know they have a problem. They're looking for a solution, and it's more about showing that your technology is mature enough for them to implement it. So, and this kind of goes back to what I think is so super interesting and great about this Mango and then the space is that normally when I look at companies, I look for having like the right ingredients. So you need to have like the market, you need to have people who are willing to buy the product. And in our case, I feel like there's a strong pull from these pharmaceutical companies because they're actively looking for a solution. And then you also need to have like a technology that differentiates what you can do. And in this case, we have the Caltech fundamental patents, and we built our patent portfolio. On top of that, that has been really strong, and also it gives us a moat and an edge. And then it really comes down to execution. So this is, as an engineer myself, like this is what I love. It's like you have the market, you have the technology, let's just go build it. So put together the right team, and as long as you execute, the future of the company is very bright.
So, David, maybe just to pick up on one of the questions that came in from the audience relating to scaling, you showed the M6 unit, which holds 6 cartridges, but the picture looked like it actually had like a daisy chain, set of M6 units numbered 1 through X.
Yes.
And the question is, why 6? But I think the real question is like, how does this scale? Since one of your advantages to other systems today is the footprint. And so how do you think about sort of the footprint and scaling when you're now handling commercial level, production, whether it's a large molecule or small molecule drug or, a cell or gene therapy?
Yeah, so we chose 6 because it's a multiple of 3, and this is really ties into sterility, where you typically have 2 aerobic test, one at 22.5 at 32.5 degrees Celsius, and then one that's anaerobic. So it really, we want to set it up for that. So we have multiples of 3. And the way we designed it is that each bay is removable and it's actually hot swappable. So you can actually take one bay out. You can run 4 bays or 5 bays if you want to. They're completely independent. And so you're able to scale this. So if you need 12, you can put in 12. If you need 10, you can put in 10. And like you said, this is a benchtop device. It's not, it's a large coffee machine, I would say. That's the size of it. So you can have these right next to each other. You can even stack them if that is what you wish. So it's very flexible in that sense. And of course, you also get the results back much faster, so the turnaround time is faster, so you don't need to have much availability as you would with a normal petri dish.
Thank you, Simon.
And then piggybacking on Al's question, and then obviously Jeff as well, they're all kind of interrelated here a bit, but just can you maybe just dig in deeper on the computer vision piece just to make sure people are tracking along with that and just what that means and how it impacts things, and that may lead to some of the discussions on what Al was digging in on too, which is the just the cost impact of this stuff. Is it, usually it's more log scale versus like incremental. So just would be curious your thoughts on that.
Yeah, so I think this is like the perfect use case of AI and machine learning. This is where you have a pretty defined data set, and you also really know what you want the outcome to be, and what you're really using the machine learning and AI to is to optimize this, make sure the algorithms catch like all these edge cases and also different types of microorganisms. So we've spent a lot of time refining models, training models, and what's even surprising to us is how quickly we're able to get very good results. And I said in the presentation that our results are on par with a human sitting manually looking at each square, zoom all the way in, and this can take hours. And the model spits the same out in a few seconds. And even though I said it's on par, that's actually not true. It's actually already better. It's currently, our scientists use it. we don't actually go in and manually check these anymore because it is that good, and it's able to pick up even the subtle contrast changes that a human eye wouldn't notice. And this means that we can get the results even faster than what we could even if you had a whole, like if you had 100 people sitting in a room and pixel peeping on every single test, they would not be able to deliver these results faster than the AI can. So that has been that's a super game changer for the industry because it, of course, is needed in order to automate this process, right? Without AI, you cannot. It's not feasible from a cost perspective to have this done manually, and that kind of brings us into how we see this scaling. Right now, the cost of each test is higher than a traditional petri dish, but of course, you get the benefit of it being super fast and you saving costs there. So overall, it's a cost saving. In the future, as we get the cost of production and the volume higher, we definitely expect this to be significantly lower than even a traditional petri dish. So even for tests that are not time sensitive, we expect that Mango will be the best solution.
And then, I don't know, just maybe paint a picture of what, and maybe even visually, if you can describe it, just like what is full scale automated biomanufacturing?
Yeah.
Like, what does that future look like? Because that's, I don't know, it's an interesting world, and I think one that we're heading towards.
Yeah. It is really where everything currently we have these like big vats. It almost looks like it almost looks like brewery. If you have ever seen like a brewery where you brew beer, you have these huge vats where you mix ingredients together. They go through different processes, and that's how the drugs are made. In the future, what you really want is you start with the ingredients, and then everything else is automated. So all the mixing, the whole process flow until the end is completely automated, and that is the goal. And then along those steps, so currently, typically it's about like 10 times doing this process. You take samples and then you run these tests, the bioburden and sterility test on them at various times, different tests, but you take these tests automatically. So we see Mango being a inline system where it automatically pulls a sample from the line and actually only runs a test on that sample and spits out the information automatically without actually wasting any drug product. So right now, so some of these products you actually waste hundreds of thousands of dollars just on getting these samples, because once you take these samples, it's actually a wasted drug product. But this is actually a good product that you could sell if you can filter this in line. So there's a big cost saving there as well.
And Simon, does that kind of imply that that pharma manufacturers and biopharma essentially need to move away from petri dishes, regardless, just because it appears to be part of sort of a manual system, and it also requires that the sample be taken out of the system, so to speak, exposed to the air, dropped, whether it's by a person or an automated pipette, it still needs to be dropped, onto a dish and then read. So is that how you're thinking about it?
And I think you could do it with petri dish, but the bottleneck that you have currently would just be so much more severe in the future. So when everything else is automated,
just the sheer time,
slow manual process, it's just going to slow the whole process even further. And it's already a bottleneck. So it's the fact that it will get worse is something that all the manufacturers are fully aware.
Right.
Yeah. Well, and one thing I just want to touch on perhaps is just the applications of this particular system, if you will. That's a high growth area for just where therapeutics are going as well. So it's like you're in a good spot because it's a massively growing market, in terms of just a modality, if you want to put it that way.
Yeah, there's many different areas where
Oh,
this
Kevin, I
is
think
a
we're
very
getting
like
feedback, just FYI.
yeah, there are many different areas where this is applicable in terms of different treatments, but also even for things like water monitoring. This is something, air and water monitoring. It's tests that are done very, very frequently. It's not as time sensitive, but this same technology can be used. We talked to manufacturers of baby wipes. They said that currently they have to ship the wipes to warehouse to hold while they get the sterility test back, and then once it's clear, they then take it out of the warehouse and ship them off. With this kind of test, they can already put them on trucks and then ship them out, and then they don't have a need for this warehouse that they were estimating costs like them tens of millions of dollars a year just to warehouse this product because you have to unload and load trucks repeatedly. So there's so many different verticals where this is super interesting. I think when it comes to pharma in general, I think we very much focus right now on the drug production portion, and then we go into the sterility portion, especially with gene and cell therapy. I think this is where we see a huge pull, and also what is interesting about that is from a regulatory standpoint, they have exceptions to really implement these new methods much quicker than other areas because there is a need, and the government also realizes there is a need, so solutions can basically become fast-tracked into production. So this is why we are also very bullish on the gene and cell therapy space.
Gotcha. Super interesting. Any other questions from the audience, from Kevin? I saw Brian had a potential business development opportunity for you in the chat. So I did put Simon's email address in the chat, in the webinar chat. So if anyone has additional questions, happy to stay on here for a couple of minutes if they do. Otherwise, questions or as Brian mentioned, maybe even an opportunity, feel free to reach out to Simon. They're doing
Yeah. And I should also mention we're based in Los Angeles. We are right in the Playa Vista area. So if you're interested, we're happy to host and show the technology in person as well. This is something that gets me super excited, so I love to show it off.
Yeah. Yeah. No, it's an exciting future. It's coming sooner than yeah. It's overdue, but coming quickly.
For sure. I think it is the right elements, the right timing is kind of coming together where the need is very much there and it's been focused. If you had the same technology maybe 30 years ago, it's not clear that you would have success. But I think the external factors really help us, and also with AI being available to run the analysis, it's kind of like a key aspect that enables this technology.
Yeah. Yep.
Yeah, I don't have any other questions, David, but Simon, thanks for your leadership. Thanks for taking the time today. David and I are both very excited about the prospects for Mango as one of our most exciting portfolio companies. So thank you.
Yeah, thank you, David and Kevin, and thank you for all the support from Free Flow. You guys have been excellent to work with, and we appreciate all the help that you give, not just from you definitely what I call value added investors. So appreciate all the hard work you guys do too.
Thank you. No, it takes a village, right?
For sure.
Yeah. 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 Just I don't think I said this at the beginning, but we were the only investor in the pre-seed into this company. So we kind of found it almost in the lab, just coming out. That's what we usually do. So but it's now going on 4.5 years or so since we've been in it, and it's been really cool to see the progress actually. So thanks again for carving out the time, and thanks to everyone who joined the conversation as well. Have a good rest of the day, and we'll wrap it up there.
That's good. Thank you.
Yeah. Take care.
Thanks, Simon.
Bye.
Thanks, everybody.
Thank you.